Your teacher today is Dr. Bbosa Science. Dr. Bbosa is a teacher at the Science Foundation College, the best school that teaches sciences. You can join it today. Telephone +256 753 80 27 09. Today, 27th October 2020, we are going to look at genetics, lesson 2 of 2 for A-level students. Dihybrid inheritance. This is inheritance of two pairs of characteristics. For example, when a tall pea plant possessing purple flowers was crossed with a short pea plant with white flowers, all F1 generation plants were tall, possessing purple flowers. When F1 generation plants were self-pollinated, the F2 plants contained plants of 4 different phenotypes, i.e., tall plants possessing purple flowers, tall plants possessing white flowers, short plants possessing purple flowers, and short plants possessing white flowers in a ratio 9 to 3 to 3 to 1. This can be illustrated as follows. If T is gene for tall plant, small t gene for short plant, capital P gene for purple flowers, small p gene for white flowers, the cross can be carried out as follows. Parent phenotype, we have tall and purple flowers against short white flowers. Then parent genotype, parent genotype will be capital T, capital T, capital P, capital P, against small t for short, then small p for white. Then gametes, here all will have capital T, capital P. Here you will produce gametes of small t, small p. Then offspring F1, we shall have this one coming together, then we shall have capital T, small t, capital P, small p, and all of them will be tall and purple. Tall with purple flowers. Then F2 generation, we shall use a Punnett square. F2 generation, then we shall use a Punnett square. Then one parent will have a gamete for capital T, capital P, or a gamete for capital T, small p, or a gamete small t, capital P, and a gamete for small t, small p. So one parent will produce 4 gametes, capital T, capital P, capital T, small p, small t, capital P, small t, small p. Another will also produce similar gametes, which is capital T, capital P, capital T, small p, then small t, capital P, and then lastly small t, small p. When these two meet, you form capital T, capital T, capital P, capital P. When this one meet with this one, to be capital T, capital T, capital P, small p. When this meet with this, to be capital T, small t, capital P, small p. When this one meet with this, we will have capital T, small t, capital P, small p. If this fertilize this one, we shall have capital T, capital T, capital P, small p. If this one fertilize this one, will be capital T, small p, small p. When this one fertilizes this, becomes capital T, small t, capital P, small p. When this one fertilizes this one, will be capital P, capital T, small t, small p, small p. When this one fertilizes this, it will be this capital T, small t, capital P, capital P. This fertilizes this one to be capital T, small t, capital P, small p. When this one fertilizes this, to be small t, capital P, capital P. When this one fertilizes this, to be small t, capital P, small p. When this one fertilizes this, to be capital T, small t, capital P, small p. When this one fertilizes this, to be capital T, small p, small p. When this one fertilizes this, small t, capital P, small p. When this one fertilizes this, we shall have small t, small p, small p. Then the phenotype, we shall have tall, purple. Tall purple. Whenever there is capital T and capital P, it will be tall and purple. So we shall have 1, 2, 3, 4, 5, 6, 7, 8, and 9. So we shall have 9. Then tall, white, that is capital T, small p. We shall have 1, we shall have 2, we shall have 3. So we shall have 3 chances. Then short, purple. Then we shall have T and capital P. Whenever there is small t and capital P, that is 1. This is 2. This one is 3. So we shall have 3 chances of short and purple. Then short, white, that's small t, small p, we shall have 1. Therefore, the probability of phenotypes will be 9 tall and purple, to 3 tall and white, to 3 short and purple, to 1 short and white. Modification of the ratio 9 to 3 to 3 to 1 above epistasis. This is a condition where one gene on a different chromosome interacts, modifies, or masks a gene on a different chromosome. For example, a gene T on chromosome 9 gives a pigment that is brown, and a gene P on chromosome 7 gives the same pigment. But when both T and P are present, they give a purple pigment. Assuming that brown is dominant over purple, the phenotypic ratio becomes, so when you fill up a Punnett square, to have a purple color you have both T and P. So purple, P and T, 1, P and T, 2, P and T, 3, P and T, 4, P and T, 5, P and T, 6, P and T, 7, P and T, 8, P and T, 9. So you have 9. To have brownie, we have either P or T. So when you have T, 1, T, 2, T, 3, T, 4, T, 5, P, 6. Then when you have small t and small p, we have 1 and 1. So the ratio becomes 9 to 6 to 1. Example 2, a gene T on chromosome 2 gives an iris Drosophila, whereas a gene P on chromosome 4 gives a brown-eyed Drosophila. So the phenotypic ratio becomes, every time you have capital T, the iris, the Drosophila don't have eyes. When you want brown, then you must have capital P, and any other color, small t, small p. So let us see how many are Irish. That is 1, 2, there is a capital T, 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 though there is no eyes. There is a capital T, there is a capital T. Whenever there is small t, the eyes are there, but with the capital P, they are brown. So this is 1, 2, 3. Then for any other color of the eyes, then small p. So the ratio becomes 12 to 3 to 1. Number 3, dominant complementary genes. This is where T and P are necessary for the color to be expressed. The phenotypic ratio becomes, so we have colored and colorless. As long as capital T plus capital P are present, they are colored. Either TP or capital PT or small TP to be colorless. So Those are colored capital T, capital P, capital T, capital P, capital T, capital P, capital T, capital P, capital T, capital T, capital T, capital P, capital T, capital P, capital T, capital P, capital T, capital P. Those that are colorless will be 1, 2, 3, 4, 5, 6, 7. So the ratio becomes 9 to 7. 5. Gene T in a mouse gives a gray coat, while gene P gives a black coat. But when both genes occur, T is dominant over P. Find the phenotypic ratio. To have a gray coat, you have capital T, or capital T and capital P. To have a black coat, you must have capital P, small t, colorless, small t, small p. So how many are gray? 1, 2, 3, 4, 5, as long as there's a capital T, 6, 7, 8, 9, 10, 11, 12. As long as there's a capital P, it is black. 1, 2, 3. Then this is the colorless. So the rest becomes 12 to 3 to 1. Gene linkage. This is the existence of many genes on one chromosome which are segregated together. Sex-linked genes are genes that exist on sex chromosome, usually X chromosome. Y chromosome has got very few linked genes. Sex-linked traits include color blindness and hemophilia. Color blindness is more common than hemophilia because hemophilia is lethal or leads to death. Sex-linked traits are more common in male than female because females have 2 X chromosomes with a possibility of a carrier state, while male don't. Transmission of sex-linked character. For the parent, the male is hemophilic, the female is normal, then all the children are normal, but the girls are carrier. When the man is normal and the woman is a carrier, then half the girls are normal, half are carriers. But half the boys are hemophilic, half the boys are normal. Sex-limited characters are characters that show up only in one sex. For example, uterus in female. You can never find a uterus in a male. Scrotum in a male. You can never find a scrotum in female. Crossing over. In prophase 1 of meiosis, segments containing genes may be exchanged between chromosomes, leading to recombinants in offsprings. The crossing over value is equal to the number of organisms with small proportion of exchanged genes over the total number of individuals. Ratio genes. This is a genotype that leads to the death of organisms possessing it. For example, homozygous genes for yellow color in rats. So if a rat is Y, Y is crossed with another rat which is YY, which is heterozygous, then we expect to have Y, capital Y, small y, then capital Y, small y, then small y, small y, small y. You find that this one was die, such that offsprings will always be 2 gray and 1 brown. So the ratio 2 to 1 represents lethal genes. Thank you so much for listening to this video. Please find related videos and other videos on YouTube channel, Digital Teachers Uganda. Go to YouTube, where there is search, type in Digital Teachers Uganda, then you'll be given categories of videos. Our website is Digital Teachers. Here you can find text and videos, notes, text, exams, marking guide on this website. Please subscribe to promote my videos. Subscription is free. Let us share these videos with our friends to increase the number of students during sciences. If you are in Chiwanga, you can buy your drugs at Mvanunji Pharmacy.